Hey everyone! Not all microbial cells look the same. Some are spherical, some are rod-shaped, some spiral like corkscrews. Others grow in chains, clusters, or long filaments. And those shapes are not random. Cell morphology can influence how microbes move, how they obtain nutrients, how they attach to surfaces, and even how they survive in different environments. In other words, shape affects function. In this lecture, we'll explore the major types of microbial cell morphology and arrangement, and how differences in shape can help microorganisms survive and interact with their environment. When microbiologists first observed bacteria under the microscope, one of the first things they often notice is cell shape. Although there is enormous diversity among bacteria, many species fall into a few common morphological categories. Cocci are spherical or round cells. Some exist as single cells, while others form chains or clusters, depending on how the cell divide and remain attached. Bacilli are rod-shaped bacteria. This is one of the most common bacterial morphologies and is found in many different environments. Coccobacilli are short, oval-shaped rods that appear somewhat intermediate between cocci and bacilli. Vibrios are curved rod-shaped bacteria. Their curved shape can influence how they move through aquatic environments. Spirilla are rigid, spiral-shaped bacteria that often move using external flagella. Spirochetes are also spiral-shaped, but they are thinner and more flexible. Many spirochetes move using specialized internal flagella, giving them a corkscrew-like motion. These shapes are not just useful for identification. Morphology can influence movement, nutrient uptake, attachment, and interactions with the environment. Bacterial cells are not only classified by shape, they are also classified by how they arrange themselves after cell division. When bacteria divide, daughter cells sometimes separate completely. In other cases, the cells remain attached, creating characteristic patterns. For example, cocci may form pairs called diplococci, chains called streptococci, or grape-like clusters called staphylococci. Some rod-shaped bacteria also form chains or align side by side, depending on how division occurs. These arrangements are influenced by the plane of cell division and whether cells remain attached after dividing. Arrangement patterns are important in microbiology because they can help scientists identify organisms and understand how cells interact with one another. In some cases, cellular arrangement may also influence how bacteria attach to surfaces, form biofilms, or interact with hosts in the environment. Cell shape is not just something microbiologists use for identification. Morphology can directly affect survival. One important factor is surface area. Cells with a high surface area relative to their volume can exchange nutrients and waste more efficiently with the environment. This is especially important for microorganisms because most transport occurs across the cell surface. Shape can also influence movement. For example, spiral shaped cells may move more efficiently through liquids or mucus like environments. Cell size and shape may even affect susceptibility to predation. Some morphologies can make microbes harder for predators or protists to capture and consume. Arrangement matters as well. Chains, clusters, and biofilm forming structures can improve attachment to surfaces and help cells remain in favorable environments. Overall, morphology often reflects environmental adaptation. Different shapes and arrangement can help microorganisms survive, compete, and function in very different ecological conditions. One of the first things microbiologists look at when examining a sample is morphology, the shape, size, and arrangement of cells. Morphology is useful because it gives us rapid clues about the organisms that might be present before we ever run biochemical tests or genetic analyses. It helps with identification, classification, and in some cases can provide an important early diagnostic clue. If you look at the micrograph in this slide, you can already see two very different types of bacteria present. The purple spherical cells are cocci, while the short pink cells are rods, also called bacilli. Now, imagine that the sample came from a patient suspected of having a urinary tract infection, or UTI. A microbiologist would immediately pay close attention to the pink rods because rod-shaped bacteria, especially gram-negative rods like E. coli, are among the most common causes of UTIs. The purple cocci may still matter, but depending on the sample source and abundance, they could simply represent normal skin microbiota that contaminated the sample during collection. This is why morphology matters. Even before we know the exact species, cell shape, staining patterns, and arrangement can help microbiologists make informed decisions about what organisms are most likely important in a sample. Although cells come in many different shapes, maintaining the shape is not easy. Cells constantly experience osmotic pressure because water moves across membranes. In many environments, this pressure can place major stress on the cell. Without structural support, cells could swell, deform, or even burst. To prevent this, many microorganisms possess rigid cell walls surrounding the plasma membrane. Cell walls provide structural strength and help protect cells from osmotic stress. They also play a major role in determining cell morphology. The shape of a bacterium is often strongly influenced by the structure and organization of its cell wall. So while the plasma membrane forms the living boundary of the cell wall often provides the physical support that helps maintain cellular shape and integrity. In this lecture, we explored the diversity of microbial cell morphology and arrangement. We saw that microorganisms can exist in many different shapes, including cocci, bacilli, vibrios, spirilla, and spirochetes. We also discussed how cells may arrange themselves into pairs, chains, clusters, and other patterns after division. Importantly, morphology is not just descriptive. Cell shape and arrangement can influence nutrient exchange, movement, attachment, environmental interactions, and survival. We also introduced the idea that maintaining shape requires structural support. Many microorganisms rely on rigid cell walls to resist osmotic pressure and maintain cellular integrity. In the next lecture, we'll focus specifically on the bacterial cell wall and explore how its structure influences protection, morphology, and staining characteristics. Thanks so much for attending this lecture. See you at the next one.